Affordability and access to medicine is a global need. Generics and biosimilars increase competition, improve affordability, and improve access to medicines. Assurance of therapeutic equivalence is critical to ensure patients experience equivalent therapeutic outcomes in the real world. Hello, I'm Ejaz Hussain, and I wish to share with you my experiential learning about therapeutic equivalence. My experiential learning of equivalence has influenced and shaped my professional development. As a graduate student in 1984, being aware of Drug Price Competition and Patent Term Restoration Act, the Hatch-Waxman Act, to my experience at Sandoz as a global head for biosimilar development when Biological Price Competition and Innovation Act of 2009 opened the door for biosimilars in the US. What is experiential learning and how does it relate to equivalence, especially therapeutic equivalence, and why it is critical to provide the assurance of equivalence patients need is a topic that I wish to discuss with you. Experience is our practical contact with objects, subjects, and events in the world outside us. Experience, verb, is our feelings when we are in contact with objects, subjects, and events. Feeling our emotions is a key to experiential learning. Experiential learning is learning through reflection on doing. Caring is research, knowledge is power, and wisdom is practice. Making space for research, knowledge, and practice over a product's life cycle within the sector, pre-approval and post-approval, with a knowledge, with awareness of the real-world consequences is a key to providing assurance of quality and assurance of equivalence. To do so, we must appreciate the importance of integrating feelings, learnings, and our science, or feeling, learning, and our design space from our own perspective and from the perspective of patients. Design space was a terminology that was defined, described in ICHQ8 guideline pharmaceutical development. It relates to our ability to describe and predict the behavior of pharmaceutical manufacturing process and products within a space that we have considered during our development. Learning space and feeling spaces are within us. They relate to how we learn from our experience. And those are important considerations which we often do not pay attention during our formal education and training, and we must leverage in our day-to-day experience to continually develop ourselves to be assured so that we can give the assurance patients and their providers need in the real world. The 21st Century Initiative for Pharmaceutical Quality at FDA sought to improve the scientific basis for pharmaceutical product development and regulatory review, to allow ourselves the basis to make risk-based decisions based on scientific data and information. I Can See Clearly Now was part of a vision statement that was articulated in 2004. That's when this slide was constructed. I Can See Clearly Now, Vision 2020, it has taken a long time for myself to recognize that learning space and feeling spaces have to be integrated with design space. And that's the story I wish to share with you. In 2014, the Gold Sheet, a trade magazine in US, had interviewed several individuals. One was Dinesh Thakur, is the whistleblower for the Ranbaxy situation, which created a lot of challenges. It was another from somebody from FDA. FDA was saying, look out for these issues in data integrity. And consultants were saying, focus on India, mask the real data integrity problem. Data integrity issues are all over, not just in India. From my perspective, being an Indian American, I saw the opportunity and the need to facilitate the young talent in India, very qualified, to really leverage their qualification to develop themselves through their experiential learning. The challenge then is what Dr. Deming has said: a bad system will beat a good person every time. The challenge to assuring quality and therapeutic equivalence in a system in which you have data integrity and other GMP violations is a major challenge. How do we build and improve a quality management system from within? Dr. Deming articulated a way forward, which is the same as what Rumi had articulated. It is a common cause strategy. We have to be the change. The premise of my talk really are twofold. One, across the global supply chain, we have not paid adequate attention to professional development. And so professional development across the supply chain is highly variable. And the second premise is we underestimate complexity and we find ourselves in over our heads very frequently. So we must align and integrate our practices of quality by design with systems approach to quality management. And we can only do so when we go beyond our traditional education and training to leverage our experiential learning. I believe it is crucial and urgent to pay attention to professional development in the context of rapidly evolving stakeholder expectations, socio-political and economic dynamics that we see around us, and increasing emphasis on real-world evidence, particularly in the US in the context of the 21st Century Cures Act, which was signed into law in December of 2016. For decades, we have struggled to meet the needs and expectations of our stakeholders. From the generic drug scandal from 1987 onwards to the current challenges in CGMP violations that we see across, the high percentage of those. We continue to make mistakes. Recurring errors are mistakes. That means we are not learning. The rate of our external quality failures is too high, particularly in the context of the experience economy and what the patients are experiencing in the real world. There is a risk we pose to patients, and the risk also includes eroding the trust they must have in our system to experience therapeutic outcomes equivalent to that of the reference product. Design and systems thinking are essential to reduce complexity through research and development and to solve systemic problems to prevent recurring errors and to minimize external failures. Although on paper we have outlined the principles of quality by design and quality management system, our understanding of these principles is highly variable, and more variable is our practice. Heart of the matter is to improve a pharmaceutical quality management system from within. We must illuminate paths for professionals at every level in every job to develop themselves to improve the systems from within. That's a fractal approach to building and improving a quality management system. In this presentation, I will focus its relevance to therapeutic equivalence. As I have alluded to before, professional development means to go beyond traditional education and training, to be self-authored, self-assured, so that we can give assurance to others. We must be self-assured, self-authored in what we know and how we know it. How we know it is a question that requires us to question our assumptions, questions our blind spots. We must be self-transforming in filling gaps between what we know and what we can implement. We need to feel the need and know the ways to leverage our collective experiential learning and understand the context of our abstract nouns. Assurance is an abstract noun. Equivalence is an abstract noun. We can do so by increasing our order of consciousness from that of a profiteer to patient to professional to good practitioner. All of these stages of development are within us. A profiteer who learns to be patient develops to become a professional. Feelings and feeling our emotions is key part of our expectation and satisfaction. Satisfaction is the difference between what we expect and what we observe. Our mind and our human nature is built up, has evolved with a prediction error, which is called dopamine reward prediction error. What it simply means is this: if we get a reward what we expect, we are not as satisfied. We are truly satisfied when the reward we receive is more than what we expect. That is a built-in more and more with less and less mindset that we have, which we have to overcome. But we have to recognize that in the real world, with income inequality, inability to afford health insurance, inability to afford medicines, we have growing inequality. The challenge we face is to provide assurance of equivalence in inequality. Let's not underestimate the challenge. The way we can do that is with integrity and to provide assurance that we don't have to step back from. We have to reduce our errors. We have to reduce our warning letters. We have to reduce our import alerts. We have to be true to what we say we can do. Process validation means internal validation and external validation. That's the way to think about in the 21st century cures with the context of real world evidence. Why different therapeutic outcomes can be observed and expected. Because of information asymmetry, therapeutic equivalence, the evidence of therapeutic equivalence that we collect internal to our sector is either not well understood or not well appreciated in the real world by patients and the providers. Pharmaceutical equivalence, bioequivalence, label, CGMPs are the four components of therapeutic equivalence. Therapeutic equivalence, by definition, is an expectation. It's not a proof of equivalence in the real world. It's an expectation. And assurance of quality, assurance of equivalence, and the trust the patients have and the providers have in our system is the bridge to assuring equivalence in the real world. All of us including patients, their providers, and our professionals, are at different stages of our development. We organize ourselves based on our education, training, and experience within a corporate organization. We have different jobs and responsibilities. We have an expectation of reward for our jobs. Within that context, we have to understand and recognize the expectations and the assurance patients need in the real world, and how our own development on how our self-assurance relates to that. And that is really what the key message is. Ideally, we have the same expectations and we observe the same outcomes, but that's not reality. If you really take a moment to reflect on the new drug development process, expectancy effects are so important that we have a name for them: placebo and nocebo effects. We take extreme precaution. We don't trust the physicians, the most qualified physicians who are doing the clinical trials. Obviously, we don't trust the patients to be objective in their observations of their experience of therapeutic outcomes. Therefore, we use double blinding and placebo arms as controls to gather evidence of, of safety and efficacy, of therapeutic benefit, and risk. In the real world, you don't have those controls. So what do we have in the real world? It is trust in our professional practices. The regulatory system in the US and around the world have evolved step by step as a reaction to a problem. From 1937, the elixir of sulfanilamide becoming a trigger to accelerate enactment of the Food and Drug Cosmetic Act in 1937, many amendments, the scandals, the disasters have added layer after layer of new laws intended to change our behavior. How do we go from a reactive system to a continually improving system is a question that we all should consider. And for generics and biosimilars, the two laws, the Drug Price Competition and Patent Term Restoration Act and the Biologics Price Competition and Innovation Act, really are the starting point for us to keep improving our system. They are not perfect. Nothing is perfect, but we can improve upon. Therapeutic equivalence, by definition, is an expectation. As I mentioned earlier, there are four parts: pharmaceutical equivalence, bioequivalence, equivalent label, and equivalent CGMP. We don't call it equivalent label and equivalent CGMP, but that's the way I would like you to think about it. But the key here is pharmaceutical equivalence is of paramount importance to ensure equivalence at the beginning of our development, beginning of our approval process. And we have not paid attention to pharmaceutical equivalence to the extent that we should have. Quality by design was the basis for improving that attention to pharmaceutical equivalence. Now, just from a statistical perspective, the J.S. Mills method of difference points to the importance of therapeutic equivalence. Because if we compare two groups, treatment and control, for example, and observe a salient contrast at the end, the difference must be due to experimental manipulation if the groups were equivalent at the beginning. Why is that relevant? It is relevant because equivalence assessment in a highly variable environment where the variance in where the signal to noise ratio is not optimal, when the products are designed to be the same, is not practical. It's all very difficult. Therefore, we rely on the scientific underpinning of pharmaceutical equivalence, bioequivalence, and our practices, GMPs, to assure therapeutic equivalence in the real world. Pharmaceutical equivalence and when necessary bioequivalence is the framework under which the small molecule generics are regulated. Bioequivalence is assessed using analytical methods that are most sensitive to detect differences in the two products which are supposed to be the same. Bioequivalence methods that we use, order of preference are in vitro, pharmacokinetic, pharmacodynamic, and clinical endpoint. In fact, the clinical studies are too blunt an instrument to measure the differences in pharmaceutical equivalent products. Just think about it. Many new drugs fail when because of placebo effect. They cannot show a difference between an active drug and a placebo. And the placebo that we use in new drug development is made identical in color, shape, and everything. But when it comes to generic, to open the door for competition, we have relaxed the definition of pharmaceutical equivalence so that the color can be different, the shape can be different, the mechanism of drug release can be different, and so forth. That is necessary to have a viable generic sector. But we have to recognize that in that flexibility are sources of failure as well as misinterpretation, because in the real world, when color and shape of tablets change and patients' adherence rate to their prescription changes, it is a canary in the coal mine that our current system is not providing the level of assurance patients need. Expectation of same therapeutic outcome that patients experience in the real world is a key driver for our quality by design and effective quality management system. It has evolved from our ability to provide assurance on average to being 95% confident for a population. But now we have to think about for each patient. Because if you look at the legislation, as particularly with the US legislation as is drafted for biosimilar interchangeability, the word same in each patient is in that law. How do we achieve that? But let's focus our attention on the perception and satisfaction patients all over the world have in generics. While acceptance of generic medications is improving, substantial mistrust and lack of confidence remains across the globe. For example, in Australia, only 6% of pharmacists considered that dry powder inhalers were interchangeable. In Europe, 89% of pharmacists reported receiving patient complaints regarding use of generic medicine, although 64% suggested that this was due to nocebo effect. Nocebo effect, we think, is in our mind. That's not correct. Nocebo effect has physiological impact. Patients actually feel it. These are real effects. So I think we have to remember this, particularly today with the geopolitical situation, with the anxiety and fear that is existing in US and across the globe. Expectancy affects the experience patients have is very, very important. We are in an experience economy, and no wonder that placebo effects are exploding. And no wonder we see expectation effects of price as a proxy for efficacy. The fundamental theory, fundamental foundation of pharmacology is dose response relationship. When you see price response relationship being reproduced in publications after publications, we have to pause and think about the challenge we are facing. So upon switching to generic, if patients do not expect the same therapeutic effect, the placebo and nocebo effects will kick in. And we are seeing reports after reports of that happening not just in US, but in New Zealand, but in Europe, where treatment outcomes with biosimilars, be aware of nocebo effect. What is associated with increased side effects and lower perceived efficacy following switching to generics in New Zealand? And more recently, in Nature Reviews of Rheumatology, I think this nocebo cycle was presented. Nocebo effects are noxious changes in a patient's symptoms or physiological conditions that occur because of patient's negative anticipation of treatment and might result in suboptimal outcomes and non-adherence. As as a sector which designs, develops, and manufactures products, our role is a small piece of this nocebo cycle, uh, because we have to provide assurance of quality. And when color and size matters, then we have to think about how do we overcome this challenge. Because if color and shape are part of pharmaceutical equivalence, we will not have many generics. That's not the way to think about that. So negative publicity has to be avoided. So in summary, I think feelings and emotions shape the experience of patients, and the therapeutic outcome that they experience is influenced uh, significantly from that. We know that from the clinical trial data of placebo and nocebo effects, but now we are seeing that in the real world also. And in the context of real world evidence, we have to be more cognizant of the importance of quality assurance and assurance of therapeutic equivalence and the need to reduce errors to ensure that we maintain the trust patients have in our system. And a warning sign, this is a canary in the coal mine, that should be a wake-up call for us to improve our systems and improve our quality by design. And I think pharmaceutical sector is part of the medical sector, the healthcare sector, and regulatory sector. So I think the anxiety and the fear and the inequality we see around the world puts a demand on us to develop and overcome the challenges that we have internally, so that we reduce any likelihood of eroding trust of patients in our system. I think I'm just showing this slide to share that this is a global challenge, not just within our sector. It is a challenge in the healthcare sector, and which all points to professional development as a key way forward. So let me talk to you about specific case examples. I had the privilege to lead the FDA effort and be the chair of the working group that translated the research that was done at University of Michigan and Uppsala, particularly, and Maryland, into a policy which we called a waiver of bioequivalence, bioavailability, and bioequivalence studies based on biopharmaceutics classification system, or BCS. So this was sort of an early experience on thinking about equivalence in a broader context. Now, I have included these slides as an appendix, so I just wanted to give you a big picture here. The key here was, I think, from a regulatory aspect, bioequivalence, bioavailability, and bioequivalence of a solution dosage form is considered self-evident. And when you have doubts and uncertainty on how relevant dissolution test is, you cannot rely on dissolution test for assessment of equivalence unless there is a in vitro in vivo correlation. And when such a correlation is established, it is usually is a formulation specific. So the whole idea was to start the discussion is when the rate limiting step in systemic absorption of a drug after per oral administration is not dissolution, it is the permeability across the GI membranes, then measuring blood levels, doing pharmacokinetic bioequivalence trials is not asking the right question, because you are essentially measuring two products which are exactly equivalent, because the rate limiting step is permeability. So that was the basis for evolving this guideline and BCS class membership of high solubility, high permeability, rapid dissolution, particularly since because we wanted to make sure dissolution in vivo is not rate limiting to absorption, was the basis for developing this guideline. I think, but at the same time, I think what this experience provided was to question your assumptions. As as we were collecting data to justify the recommendations in the first guideline, we start seeing many examples where you saw this. For example, in one submission, new drug submission, at atenolol blood levels, you're seeing a tablet as twice as bioavailable as a solution. How can that be? And the excipient effects, particularly in this case, the excipient sorbitol, and how a low permeability excipient, a high solubility and low permeability excipient can create osmotic pressures and change the transit times, and how can was not very well appreciated. And therefore I think that points to that underestimating complexity is something that we must avoid. So, so in that sense, I think uh, quality by design and features of quality by design is to do things consciously, and not relying on just one test. And looking at it, a totality of evidence was the concept that quality by design brought forward, because there were many errors that were occurring. For example, with cyclosporin, FDA had to recall a drug product because a microemulsion was judged to be pharmaceutically equivalent to a solubilized solution, and the label indication was to give this drug product with apple juice, but the company did the bio study in chocolate milk to 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 identify to find a passing result. And they cheated, obviously. But then what we had to realize then was, until 2009, the laws allowed for a generic company to submit one passing bioequivalence study, and FDA did not have the authority to ask whether this is the right study. And they could pick and choose. If they had done 10 studies and 9 failed and 1 passed, they would just submit the passing study, and that undermined the credibility of the entire system. At the same time, I think we were considering subject by formulation interaction aspects, where some subjects would be more sensitive to certain products or excipients, and here you could see a subject by formulation interaction for sorbitol and sucrose that can be. So these were some of the questions that we have to think about in that sense. So let me bring the point that I mentioned earlier. We are underestimating complexity, and to address complexity, we have to match that complexity in the external with world with the complexity of our mind, so that through professional development we can tackle and reduce complexity, and reduce it in a way that good practices will work for a complicated system. And we have to think from that perspective. So I've chosen 4 examples. A more recent example of a launch of first generic version of a generic version of a Nasonex by Apotex, and the Omnitrope submission for follow-on protein, which is a growth hormone in US, generic enoxaparin. And something work I had done at Philip Morris is to develop a modified risk tobacco product, which is called ICOS, is evidence of reduced risk of smoking, and tobacco harm reduction was something that I just mentioned. Equivalence assessment or comparability assessment, often we only think about with a reference product, side by side comparison. that is an important aspect. But we have many times situations where uh, equivalence or comparability assessment has to be done with prior knowledge. So the work done at Philip Morris was really without a reference product in that sense, because if somebody continues to smoke, the risk will keep increasing, as shown by the red arrow. If you quit at some point, then the risk will start reducing. So there is epidemiological evidence in the real world on that. So that's the prior knowledge that exists. So if you design a product which reduces the risk of tobacco related diseases, then it should be comparable in some way or form to the cessation data that we have. Can we can we provide that evidence was a key challenge. So this was one of the most complex development projects that I was involved with, and to show and bring totality of evidence to be able to show harm reduction with the prior knowledge that exists in the real world is a case example I just wanted you to be aware of, because I think we will face many of these challenges as we go along. Now, growth hormone Omnitrope was the first follow-on biologics that was approved. It's not a generic, but I think the press called it a generic. It was not. But the challenge here really became a situation which points to integrating evidence from all parts, from CMC to farm talks to clinical pharmacology and so forth, and to make a totality of evidence decision, which is very difficult in our sector, primarily because we are so used to making a decision on the pivotal clinical trial, two well controlled clinical trials for each indication. That has been the foundation on which we make decisions on, and it has been easier to do that. But now we have to think more in systems way, and all the pieces of evidence have to come together, and we have to think about a evidence architecture. How do we arrange evidence, and how do we ensure congruence of that evidence from multiple disciplines and over time, so that the evidence of equivalence comes out as a total package rather than one clinical study or one pivotal study. And this is an important consideration, and the challenge, the solution here is to appreciate complexity. And what I see around the world is we underestimate that complexity, and we rush to clinical studies. We say most of the development projects are organized in a time to clinical, time to phase one, but we don't take the time to do the research, to get the analytical characterization done, and to design processes that allow you to be similar exactly within the variability of the reference lot to lot is sort of the key thing that we have to think about. And the challenge then was, I think, pharmaceutical equivalence is complex, not appreciated, and one must do research to develop analytical methods, the right analytical methods to understand the critical quality attributes of the reference product way beyond the compendial aspects, and to be within the lot-to-lot variability of between the lot-to-lot variability of the reference product would be one way of thinking about that. And at the same time, that allows you to overcome blocking petitions by the reference product sponsor to maintain their market share. So follow-on biologics, biosimilars, are complex, and the regulatory system that we have differs from that of small molecules. As I mentioned earlier, we have a biological license application where the process is the product. So the facility has to be licensed. And so we are dealing with biotechnology, materials, and stability of the genes, and the become very, very critical. So the residual uncertainty is very high. And the biggest challenge we have in biosimilars is not clinical evidence, it is pharmaceutical equivalence. pharmaceutical equivalence is the Achilles heel in assessment of similarity and interchangeability of biosimilars. So exquisite characterization and orthogonal methods to characterize becomes a key to this chapter. And I think with the enoxaparin, it was, although considered in the generic world in the US, it is a biosimilar in Europe. Um, it's thousands of species of carbohydrate from a natural source, starting from heparin, which comes from the lung tissue of pigs. And so here also, I think the key then became care is research, knowledge is power, and wisdom is practice really becomes is to create new prior knowledge. New prior knowledge simply means is the originator product was developed 10, 15 years ago with those analytical methods and has proven to be safe and efficacious based on clinical evidence and then the historical marketing data, pharmacovigilance data. Now we are now developing this product 10, 15 years after that with new analytical methods. And so how does one create evidence architecture to show that you're within the variability of the reference product and you have identified all the critical quality attributes, and that your manufacturing process will reproducibly and repeatably produce that product becomes a key. And the only way you do that is to truly appreciate that this is complex and make the space within your organization, within your project planning, to take time to develop your analytical methods, characterization, before you begin the process of developing and doing the uh, collecting the evidence based on pharmacology, toxicology, and bio studies that may be necessary. I think the scientific consideration, the review and approval of generic enoxaparin is a wonderful paper written by FDA, which outlines how this process evolved. And I would appreciate if you would take a look at that. The other consideration that you need to think about is the variability that you measure in the reference product 10, 15 years afterwards. Can They have been improving that manufacturing process. So it is an underestimate of the variability, which is what we show as. And especially when you're dealing with API, the number of lots of API in the lots of product may are not known to you, and therefore the right sample size of how many lots to test is important. But at the same time, you have to make sure that you have a defensible statistical analysis and acceptance criteria, because the challenges will keep coming, because with new analytical methods, people will pick up the samples from that and compare and keep claiming that your product is different, it's not the same. And so how do you overcome that becomes an important consideration. But within that research domain is also an opportunity, because when a new product is approved, it tends to be more variable, and over time the variability is reduced. And here is the quality target product profile for new biosimilars. The approach is to characterize the reference products early and many lots, so that you can achieve that. Now, at Sandoz, we invested in developing this capability in-house. But the point here is, if every generic company has to do that, that's a lot of duplication. There's a lot of many generic companies cannot afford that. This is an opportunity for the academic, other to step in to be able to do that. And this new prior knowledge then becomes available to all generics. Perhaps the compendia will pick this up so that they can do this, or academics can do this. But I think we need to find a solution that every generic doesn't have to do this again and again, and to reduce the cost and time for development. I think the key learning from the more recent one is that the right analytical method is the key, and and how you present your information to the regulators is the key. So in 2016, FDA published this FDA embraces emerging technology for bioequivalence evaluation of locally acting nasal spray in lieu of clinical endpoint study, which was deemed unacceptable by FDA. So, in case of nasal products, the site of action is the local epithelial tissue, and measuring the blood levels is not at the site of action, so it is not the conclusive evidence. So the criteria for equivalence assessment of nasal sprays or locally acting drug product is multitude, is a totality of evidence criteria. You have the in vitro from the plume geometry to particle size distribution, and at the same time then you have a PK measure to look at the systemic absorption. Then in this particular case, there was a clinical study which was the nasal drip walk in the park clinical study to have comparison. The company had rushed to the clinical study and done the clinical study, which passed. So equivalence was established in the clinical study, which is a very expensive clinical study. But the PK study failed, and the particle size was very different between the reference and so forth. So the company had to go back and to reduce the particle size and to reformulate and to show passing PK study. But then FDA had demanded the repeat of the clinical study. Now, the original clinical study was done where the particle size was clearly different, but the clinical equivalence was established. The clinical study could not distinguish between the two particle size differences. So the argument then we had with this scientific dispute resolution was that there's no need to repeat the clinical study. So in the deliberations that occurred, this review went on for 8 years, and towards the end in 2016 there was a scientific dispute resolution. That's where I got involved. So we looked at the whole thing and said, why would you need to repeat a clinical study? The answer came out is, look, one of the major side effects or adverse effects is nasal bleed, and we need to ensure that the particle shape is consistent throughout the product's life cycle, and that's a critical quality attribute, and you have not measured that. Luckily for the company had used a Raman directed spectroscopy for particle size analysis to distinguish excipients from the drug. The same methodology actually had the shape information, and therefore that became a reason for strengthening the evidence of therapeutic equivalence on the shape of the particle, and for approval of this. The key lesson that we can learn from that is good methodological evidence cannot be complex. Um, Therapeutic equivalence is an expectation and a system. Pharmaceutical equivalence is the key, which we often underestimate and is part of totality of evidence. And good material evidence should be self-evident. So I think what you're seeing here is reducing complexity to be good practitioners in a complicated aspects to make sure the evidence that patients will see is self-evident. I think that was a key lesson that helped shape my learning in this case. So in that sense, let me bring this discussion to common cause. Satisfaction in adherence is a development need. Competence, complexity, and culture, I think, is a theme that I've talked to you about. In the real world of 21st century cures, we are trying to improve medical care, reduce errors in medical practice, and as we go towards personalized medicine, patient-centric medicine, real-world evidence, I think what we are seeing is the healthcare system has to improve the quality. They're being incentivized by pay for performance, outcome-based reimbursement, and adherence to prescription by patients is a increasing need of attention, because from 30 to 50% of patients do not adhere to prescriptions, and that is a major challenge. On our professional side, I think pharmaceutical quality, we have to work towards availability, adherence to CGMPs, and affordability of our development cost and manufacturing cost is important, but we have to give assurance to patients. So assurance to patients is related to their adherence that evidence has shown now. And therefore, I think in some sense, adherence to CGMPs and adherence to prescription is a common theme that I think we can think about. Profiteers becoming patient to professionals and so forth is a common development scale that one can use. So all measures tied to the highest level of objective, I think, in terms of pharmaceutical quality management system, suppliers to process to customers, process management and improvement, benchmarking, supplier performance, input measures, process changes, process employee measures, output. All of these have to be related to quality metrics, and quality metrics that are being evolved by FDA deal with complaint rates, and out of specification, invalidation of out of specification results, and so forth. And I think those metrics are important. But the key then is patient satisfaction. There are multiple components, multiple aspects of patient satisfaction, and it deals with interpersonal and social expectations. The healthcare quality um, uh, theory demonstrate that the principal outcome is the interpretation of process of care, and the expectancy value theory, which says that personal belief and values about care as well as prior expectations about care are important. Multiple model theory, which says that expectations were socially mediated, reflecting the health goals and patient and the extent to which illness and healthcare are violated. Personal sense of self is important there, and so forth. So what it points to is the feeling, learning, and space that is so important and has become so important in experience economy, particularly with the social media that we have, that that I think we can relate to that as patients do. Our scientific education and training is obviously different from those of providers and those of patients. We will always have information asymmetry between us, but we have to maintain trust between us. And the way to maintain that trust is to appreciate the perspective of a patient, the perspective of providers, and we can do that because we have the common fractal dimension of I call it satisfaction and adherence in our procedures as well as to prescriptions. And I think that binds us together and puts us on a common development path, which I simply put it is profiteers becoming learning to be patient becomes professional. And that lines up very well with the constructive development theory of adult development of orders of consciousness, being an imperialist to being socialized to being self-authored to be self-transforming. In the context of a very simple GMP model for that is we have to go develop ourselves from looking good to being good to doing good to seeing good in everything we do. And that really brings back the message, which is very, very important. I think is a quality scorecard. I think the National Institute for Pharmaceutical Technology and Education is sort of evolving a scorecard, and you will hear much about that from Professor Steve Burns and Gary and others. But the patient satisfaction index really becomes a common theme in that scorecard. I think would be a message that I would like to emphasize. Equivalence and inequality is a major challenge, and we have to bring forward a patient satisfaction index. And that patient satisfaction index is our satisfaction is a professional satisfaction index. It's all on the continuum. And our quality scorecard really then becomes a way to guide our professional development to be able to keep up with the assurance of quality and therapeutic equivalence patients need, recognizing that profiteer, patient, professional, and good practitioners are are within the same within us and are on the same scale of a quality scorecard. So I think in a social media, likes and loves, hearts, and smileys and blue ribbons are examples of that scorecard. And clearly, I think we need to minimize errors, dangers, anything that creates anxiety and fear within the sector is something we have to work to prevent from occurring. And for that, We need quality by design and systems thinking to be aligned. Quality management system requires us to build it and improve it from within, and to do that from within, the only common cause that we often forget is us. We can only improve a system from within when we improve ourselves. So continual improvement of a system requires continual professional development. I hope that makes sense to you, and I thank you for listening.